So thank you very, very much. It's an honor to be here. I want to thank Mr. Kavli, the Kavli Foundation, the Academy, the Ministry. It's an honor and a wonderful thing to share in this celebration of neuroscience by the Norwegian people and by people all over the world. And I want to tell you the story of our work as an adventure story, because that's actually what it's been. That's what it still is. And I myself never ever would have thought when I started out that I'd wind up with a group of people working on habits and rituals and how these might relate to problems in neurology and psychiatry. After all, our habits are so familiar to us. They're so common in our lives that we do them almost without thinking, from taking the same way to work or whatever. And these, of course, are our individual habits. But we all have social habits and even societal habits. And these are somehow, to me, like rituals running through the thread of history. You're seeing here the depiction of one from ancient Crete, the Minoan civilization, maybe 2000 BC. And as we all know, these rituals can have great power when they become cultural rituals. And actually, these rituals are related to a deep issue in neuroscience, in the study of brain and behavior. And that is, how can we intend to do something and then actually do it? It's almost like a miracle. How do we develop routines and habits that become so familiar that they're comforting, even by their sheer familiarity? And this whole issue was long thought to be really run by this huge neocortex we have. This is an early human scanning picture. Here's our big frontal lobe. And the great surprise here has come that a lot of this depends on the deep brain. And I'm showing it here. It's a very special brain scan. This is one of the very first fluorodopa scans showing the dopamine, the neurotransmitter that's lost in Parkinson's disease. And this region in pink, that's the striatal. And here you can see that loss in the brain of a severely Parkinsonian patient. The dopamine is gone, and with it, there becomes a profound inability, as Parkinson himself said, to translate will into action. So the question I want to talk with you about is, how can this deep brain, thought to be so primitive, have such powerful, powerful control over our behavior? And at least part of the answer is that the brain has played a trick on us. In fact, the basal ganglia here, of which the striatum is a part, they do have descending connections to the pattern generators, a little like what Corey was talking about. Sten Gilder and his group figure these go back maybe 500-600 million years of brain evolution. But the basal ganglia also have these ascending projections, and amazingly enough, a lot of them are to the very frontal part of the brain that we think of as having high executive control. So the question is, again, what on earth could this primitive region be telling to our frontal neocortex? This is automatically advancing. That's interesting. It's telling me maybe to hurry. So I want to tell you, just to get going, that this issue has become very central in neurology and psychiatry. And the reason is that we now know that the basal ganglia are implicated in many disorders, not only the classic motor disorders. And by the way, these now we know often have emotional and cognitive signs, but also neuropsychiatric disorders, OCD, obsessive compulsive disorder, mania, depression. And many of these disorders have as phenotypes, aspects of overly fixed or overly flexible behavior and thought. So, as I'll explore to you, it's been an amazing thing for the field to learn that many of the same pathways that are implicated in these disorders are the very same pathways that we use when we learn habits and when we fall into addiction. Now, I hope you can see this picture, that it's dark enough. You're looking here, can you see it? At a cross-section through the brain of a simple rodent, just a little rodent. And you can see here, every little dot shows you where a neuron is, every little white dot. Look at this beautiful neocortex, even on a little rodent, with layers all differentiated. And then this ball of cells down here, this is the striatum. It looks crude, undifferentiated compared to the cortex. This is the view with classic anatomical stains that you and I know so well. But when we stained sections for neurotransmitter distributions, we saw something more like this. The striatum didn't look undifferentiated. It had beautiful chemical compartments. You can see them here. So instead of looking primitive, it didn't at all. And moreover, we actually first found this structure in the human brain. You're looking here at the striatum of a human, these beautiful variegated zones. We call these little pockets of low cholinergic activity. It's a cholinergic stain, striosomes for striatal bodies. And we were stunned at the time, Cliff Ragsdale and myself, as we did these stains. And then, with a series of very special students and postdocs who came to the lab, we found that this kind of compartmentalization held for virtually every neurotransmitter and neuromodulator that we could detect. It held for the inputs of the striatum, the outputs of the striatum, the birthdays of the neurons that move out during development to create the striatum in the first place. And we learned that early in development, these zones correspond to the famous dopamine islands discovered by the Swedes. The question was, what on earth could be the function of these zones? So at the time, of course, there was much excitement about the sensory maps that we have in our neocortex. And so Raffi Malek and Alice Flaherty and Hemi Parthasarathi and I decided to do experiments. We would map here the body surface. Then we would put anatomical tracers in various parts of the representation. We'd look at the tracers and find out what happened down in the striatum. Here are the striosomes, but here is a patch out in that huge matrix around the striosomes. In fact, what we found was, yeah, there are striosomes, they're chemically coded, that's very exciting, but there are patches all over. This is, in effect, a three-dimensional mosaic. You see that here in a cartoon in 2D. And in fact, what we found through many such experiments was something pretty curious, which is the cortex sent its outputs down to the striatum, dispersed them, only to have them collected again at the next stage. So we looked at this, and it reminded us of a learning architecture that was just being worked out in computational labs, including one at MIT, a learning architecture called an adaptive mixture of local experts. So what we thought was, gosh, maybe what's happening is the neocortex has a problem, okay? So it sends its outputs to a bunch of little expert networks in the striatum. Here they are in green. And then they're collected in the output stage. And here are little striosomes for all the world sitting there as though they're going to bias the system. And I'm going to come back to that toward the end of the talk. Now, we saw this input-output architecture just as Ranulfo Romo and Wolfram Schultz were discovering that this dopamine, the one in the tract that dies back in Parkinson's disease, that carries signals related to reward. So then, very shortly afterwards, with Minoru Kimura's group, we found that the cholinergic neurons that we'd been staining for, they had signals related to reward. So we thought, could it be that what the striatum is a little learning machine right down in the middle of the basal ganglia? Then experiments came along in other labs showing this is really a cost-benefit system. You can't have it just rewards in life. And in fact, this brought us very close to neuroeconomics, didn't it? And so we know now that if we are in love, we have to think very carefully, don't we? It's a different world, a wonderful one. So you can imagine now, imagine you're looking at one of these loops, it goes round and round and round until finally the activity begins to settle. Can't you imagine that maybe if that happened, you might develop a habit? And so that basically connected with a large series of experiments that psychologists had been doing that we only learned about rather lately, and that was that they had already begun to think that the striatum is important for habit learning, and they had also shown that addiction relies on it. So we decided we'll get ourselves a psychology T-maze. We set it up so the little animal, like a mouse or a rat, he sits here, a gate opens, he runs down the maze, he hears cues that tell him if he does it right, he turns the right way, he'll get chocolate. They love chocolate just like we do. It's interesting. Maybe it's a Corey innate taste. So here we have this situation. We've got electrodes in the sensory motor striatum, and we take them to school every day for a couple of hours in the lab, week after week. As they learn this behavior as a habit, and as they are learning, we're recording, finding out how does that activity change. And so let me show you that in this diagram. And again, if anyone knows how to make this not proceed automatically, tell me. Here is the run time across the top. Here's warning cue, the gate opens, they start all the way to getting to the goal, getting the chocolate about here. And I'm going to show you the training days down here. And wow, and the color shows the amount of neural activity. Red means a lot. And here, this first part shows you what happens to the activity during the whole time of initial training on this maze. And basically what you can say is, gee, there's a lot of activity, which is just fine. But look what happens as the animals continue to be trained until they really get that behavior as a habit. What we found was that the activity in that whole middle part of that habitual run, it just about goes away. And instead we were left with a lot of activity at the beginning and the end of the runs. What could that mean? Well, what it suggested to us is that maybe one of the things the striatum does is if we repeat an activity and it's good and it's rewarded, then what we do is we bracket it. We mark it in the nervous system at its boundaries. And if we do that, then maybe, again, I'm going to try one more time to turn off automatic, then maybe the way to think about this is that as we go through the stream of behavior, picking out one after another behavior to try out, we bracket the ones that are most successful in that behavior, and through that we develop a sort of behavioral chunking, a little bit like what George Miller used to talk about from memory. We remember a whole phone number, a whole PIN number, rather than every little digit. It's a way to make memory manageable. Maybe it's a way to make behavior manageable. And so we've now done many experiments, and others have too, and the upshot of these is that not only do these action boundaries form as we learn habits, but time is embedded in the representations. That dopamine modulates the plasticity. It's necessary for it. And the circuits in the striatum become linked to other circuits, probably through oscillatory activity. So you might be thinking, if it's all this goes on when we make a simple habit, how on earth could we ever break a habit? It's pretty hard, isn't it? In fact, if they were to break, would we lose those representations? And so Kyle Smith and I decided to take that up as an issue. We started recording in the prefrontal cortex and the sensory-motor striatum at the same time to find out what happened. Again, teaching a maze task, only it had a wrinkle. We call this wrinkle the bad restaurant experience. So poor little guys, we give the rats, we train them, and then we give them an experience with their reward that would be like us going to the bad restaurant, and then we bring them back the next day and put them in the maze and see, in effect, if they'll go back to the bad restaurant. If they do, they've got a mighty strong habit. So we looked at the activity, and let me show you the surprise we found. Here's this new set of animals. Whoa, stop it. Here is the the sensory-motor striatal pattern. Again, activity all over, then that bracketing pattern forms. But look what happens in the prefrontal cortex as learning proceeds. Almost nothing. In fact, it's a rather miserable pattern. We were very disappointed. But then late in overtraining, all of a sudden, the bracketing pattern occurs. So then we did the bad restaurant manipulation, called devaluation technically. And what we found was the activity pattern in the striatum is as solid as a rock. The bracket just stays. But in the neocortex, the frontal cortex, it's fickle. The pattern goes away. So it looks like when a habit is really, really strong, the striatum holds on to the pattern of that habit, even if value goes away, but the neocortex can let it go. And so Kyle and I said, well, why don't we try to manipulate that little piece of cortex and see what happens? And so we used the new technique, or then new technique of optogenetics. We put, by means of viruses, little molecules that could inhibit the cortex. And with this technique, if you shine the light, you can turn the inhibition on. And so we did that, and here's a diagram of what we found. Here is time, over experimental time. Here on the y-axis, the amount of habit. Here we've got animals, you can see their scores here, with a very high level of habit. And now we're going to shine the light. And what we found, by measurement, is that the habits, that the habitual behavior was gone in a flash. And when I say a flash, I mean a few seconds. And this effect could last for weeks. Then we turned the light back on one day, and back popped the habit. We were stunned. I'll never forget it, and neither will Kyle. We've seen this regularly. So what do these experiments suggest? To us, they suggest, first of all, even if we break a habit, its neural representation isn't gone. Somehow it's there, and we know, at least in part, it's there in the striatum. And this reminded us, of course, of what Pavlov said years and years ago. You can't forget a habit. What you do is you inhibit it. That's not necessarily good news. And just as surprising, and very surprising to me, when we have habits, we do our behavior almost automatically, just off being totally automatic. And yet there's something up there in the cortex, in the prefrontal cortex, somebody monitoring things, has to give permission in order for that habit to occur. Very strange, very, very interesting. But of course, a very interesting also question is, who in the first place is responsible for deciding it's worth packaging the behavior? And so for this, I'm going to come back to those striosomes, because we found with Frank Hebelin some years ago that these two red regions in the prefrontal cortex have the special attribute of projecting very strongly to striosomes. And we knew those regions because at the time we were finding this, the early scanning results were coming out relating these in humans to emotion. They were abnormal in people with obsessive compulsive disorder, absolutely abnormal in craving addicts. And so we decided to record and then microstimulate this little zone, the striosome projecting zone, to see what we could find. And we did this with Kenichi Yamamori. And what we did, we used an experimental protocol used in humans. It's called approach avoidance. It's used to study emotional decision-making in people. And in this system, the person or the subject is given an offer, and the offer has some good and some bad. And so you as the subject can decide either I'll take both or I don't want either one. And in the case of our little monkeys, these were lots of good food and a certain amount of air puff, irritating air puff to the face. Take it or leave it. And the interesting thing was that by using this technique, protocol, we could actually calculate a decision boundary. Here's how much air puff, how much reward. The squares show you avoidances, rejections, the little pluses, the approaches. So we could actually get a measurement of a value-based decision. And what we're finding, these experiments are still ongoing, either microstimulating the little striosome projecting zone or down in the striatum itself microstimulating, what we're finding is that we can really change that decision behavior. In the striatum, we find a mosaic of zones. Some of them make the animals more pessimistic, overvalue negative potential outcome. Others make them more optimistic. So we think now that this system probably does have something to do with the choice of one behavior or another worked out through an evaluation sidekick to the main loops. So I want to end by saying that we have been trying to get at some molecular basis of this sort of experiment. We were lucky to discover with Haruaki Kawasaki two striatum-enriched genes. Here's one in striosomes, here this orange one in matrix. They're interesting in and of themselves, but what's especially interesting to us is that Jill Crittenden came to the lab. She made a knockout of the orange gene, thus elevating the relative kind of strength of striosomes. And in these mice, we found they're repetitive, they're too focused, they tend to have stereotypical behaviors. And then to our great interest, as our work became known, we found that there are natural knockouts of this gene. And here is a basset hound who has a natural knockout of CalDF1. He's wearing, you'll notice, booties. Maybe dogs in Norway wear booties, but he's from upstate New York. He is so stereotypic, overly focusing his behavior, that when he goes outside in the winter, he burns his feet. And they have to therefore protect them. So we think we're getting close now to identifying the mutation in the gene. We think it's this little black region here. We're working on this very hard. And we also know that these genes are dysregulated in Parkinson's disease, in Huntington's disease, and that in all of these disorders listed here, the striosome matrix organization has been found to be disordered, either in our lab or often in other labs. So we're very, very hopeful that some of what I've told you might translate to do good for people. But that brings me back to the original question of these pathways attaching value to our actions. And I've shown you that there is remarkable plasticity in this system. that action boundary signals form, that we can chunk valued actions, we can manipulate decisions, either genetically or by microstimulation. And so what's important is that these are circuit level issues. This is a level that we all are hoping will help clinically and in improving our human condition. There are many people who have done wonderful work. We've had a great time in our lab. We're still having a great time. But I want to say one thing to the students here, and that is that, science, pure science, is a tremendously important calling, I would call it. A calling that it's exciting, it's very difficult, but it's one of the greatest rituals that we have in our society. And this, of course, was the inspired message of Fred Kavli and of his foundation.